this episode is about protoplast isolation, fusion and plant regeneration from them. Scientists felt the need of protoplast isolation and fusion some 3 decades ago when Cocking isolated plant protoplast. The question is, why do we need protoplast culture? That is because in a plant cell, nucleus is not the only genome site. There are genes present in other organelles like mitochondria and chloroplast. But when a male cell fuses with a female, it's only the nucleus that fuses. And so the traits present on the mitochondria or chloroplast cannot be fused. And for the varietal improvement of crops, if we want to include the traits which is present in these genomes like mitochondria or chloroplast, we have got to have a method in which protoplast, the whole protoplast of male and female, could be fused. And that is where the story of plant protoplast fusion started. In India, there are 3 laboratories working actively in this field. One is TERI, where Dr. Pentel's group is working. Another is Dr. Sen's group at Bose Research Institute, Calcutta. And the third is IARI group of Dr. Chopra and Dr. Kirti. Now for this episode, I am going to request Dr. Painter to introduce you to the subject.
Unlike animal cells, plant cells have a rigid cell wall around them. In 1960, Ted Cocking at the University of Nottingham isolated protoplasts from plant cells by removing the cell wall with a crude mixture of cellulose degrading enzymes that were isolated from a fungus. You can see in the figure how protoplasts look like. Once the protoplasts were isolated, it was found that protoplasts have some very interesting properties. They can be made to fuse and they can also take up macromolecules and virus particles. But a very important question about protoplasts was, are they totipotent? Before the work on plant protoplasts, it had been shown that somatic cells of plants can be regenerated into complete plants. Therefore, it was known that somatic cells are totipotent. That is, they have all the genetic information to give rise to a complete plant. About 11 years after the first protoplast isolation, 2 Japanese scientists, Nagata and Takebi, were able to show that protoplasts of tobacco can divide in culture to give rise to small cell colonies, and these cell colonies can be induced to regenerate complete plants. Since 1971, the techniques of protoplast isolation, the culture of protoplasts, and their subsequent regeneration have been systematically improved. Today, it is possible to regenerate complete plants from protoplasts of very diverse crop systems like rice, wheat, and soybean. Coming back to the exciting features of plant protoplasts, I have already mentioned that protoplasts can be induced to fuse. This can be done using a variety of chemical treatments like treatment with polyethylene glycol or calcium chloride at alkaline pH. Protoplasts can be also fused by electric stimuli. Another interesting property of protoplasts is that they can take up foreign DNA and therefore they can be used to introduce foreign genes into crop plants. In this presentation, we shall be concentrating on the use of protoplasts for fusion studies. Let me first show you some somatic hybrids that were made with model plant systems like tobacco and petunia. In our laboratory at the Tata Energy Research Institute, we are using the technique of protoplast fusion to make somatic hybrids of Brassica species, a group of plants which include vegetable crops like cauliflower and cabbage and major oilseed crops like rapeseed and mustard. Before we show you the procedures of protoplast isolation and culture, it is important to understand the genetic consequences of somatic cell hybridization. Many of our crop plants suffer from diseases and pests. The wild relatives of crop plants carry useful genes for disease and pest resistance. Plant breeders spend a lot of time in transferring these useful genes from wild species to crop plants. However, many times this transfer is not possible because sexual hybrids cannot be made between these due to sexual incompatibilities. In this case, somatic hybrids can be made as protoplasts of any two species will fuse. There is another dimension to somatic cell hybridization that I would like to discuss. In plants, while most of the genetic information is located in the nuclear genome, some very important characters like photosynthetic efficiency and characters like cytoplasmic male sterility are encoded by organelle genomes, that is, chloroplast and mitochondria. If we cross two plants sexually, the organelles, that is, mitochondria and chloroplasts, in most of the crop plants are inherited from the female parent. But in somatic hybrids, you make a mix of the organelles of both the parents in the hybrid cells. As the hybrid cells regenerate into complete plants, the organelles segregate out, providing us novel combinations of the nuclear and organelle genomes. Some very interesting genetical work on somatic hybrids has shown that the mitochondrial genome and chloroplast genomes can also recombine in somatic hybrids, thus giving rise to totally novel organelle genomes. You can see a comparison of the sexual and somatic cell hybridization in terms of organelle genetics in this figure. This figure shows a comparison of the genetic consequences of sexual hybridization and somatic cell hybridization.
Here we note that the possibility of getting more mutants and variants is greater in the case of somatic cell hybridization. We shall be presenting somatic cell hybridization under three headings. One is protoplast isolation, the second one is protoplast fusion, and the third one is plantlet regeneration from fused protoplasts. The technology that is available today offers two methods of protoplast isolation. The first is the mechanical method. This is an old method started by Klusker in 1892. The yield of protoplast by this method, however, is very low and therefore is not much in use today. The other is the enzymatic isolation method, either by direct or the one-step method or by the sequential or the two-steps method. Protoplast has been mostly isolated from leaf cells, callus cells, cell suspension cultures and pollen. What you are watching is the isolation of protoplast from hypocotyl. You can see here germinated seedlings of brassica from which hypocotyl is being cut and separated. These hypocotyls are then finely chopped into small pieces. The whole operation takes place under a highly aseptic condition. Excess water is removed from the sections before the enzymes, that is, cell wall degrading enzymes, are added to them. The finely chopped segments are then placed into the cell wall degrading enzymes. Usually, this solution contains a mixture of one or two types of enzymes, about 13% sorbitol or mannitol, and pH is adjusted to 5.4. They are then properly sealed and left overnight, that is 15 to 18 hours, for incubation at 25 degrees Celsius. After the process of incubation, the incubated hypocotyl segments are then very gently teased to liberate the protoplast. This digested cell, that is, cell wall's digested cells, are then filtered through a fine wire gauze to remove cellular debris. They are again washed. The filtrate is now taken into centrifugation tubes. The filtrate is now carefully sealed under aseptic conditions. The tubes are now placed into the incubator. They are centrifuged at 100 G for 1 minute. This is to remove the supernatant. The centrifugation tubes are now removed from the incubator. The pellets that we get at the base are the protoplast. The supernatant is now removed. The pellets are then resuspended in the solution. there are 3 such repetitions of resuspending the pellets and centrifuging so that we get rid of all soluble material. Finally, the pellets are suspended in 20% sucrose. The clean protoplast now floats on the top. It is picked up by pipettes for further fusion or regeneration work. Isolated protoplast starts regenerating a wall around it within few hours after isolation. Therefore, the fusion experiment is done as soon as possible. Fusion can be done with the help of chemicals, or sometimes the mechanical method or even the electrical methods have been used. Fused protoplast can be cultured in suspended liquids or in microchambers like the hanging deep method or by placing them on a solid medium, as you see these agar on the agar plate. These protoplast now develop into small embryos, and one can even see plantlets generating from them. We may derive the following conclusions. Isolated protoplast is totipotent. Protoplast can undergo intra- and interspecific fusion. Pinocytic property of protoplast makes them an excellent material for studies of genetic engineering and cell modification.